Hi everyone, thank you for tuning in to this presentation. I'm talking today about my PhD project, which I did through the Australian National University, and I was based most of the time at the Royal Botanic Gardens, Victoria, and I was studying this wonderful genus of Australian orchids, Prasophyllum, or the leek orchids, so called because they look nothing like leeks, as you can see from this photo. But they're a really cool genus of orchids. They're the third largest genus of Australian orchids with 140-ish species. They're terrestrial orchids. There's a few species in New Zealand and a few in southwest WA, but the real centre of diversity for Prasophyllum is in the southeast of Australia. And more than other orchid genera, they seem to have diversified most on quite fertile soils. And fertile soils in the southeast of Australia have been preferentially cleared for agriculture over the last 150 years. So it's no surprise that there's a lot of threatened leek orchids. We've already lost a couple of species to extinction, and there's currently 38, soon to be 39 species listed on the EPBC Act, which is Australia's national endangered species protection law. And that makes Prasophyllum the fifth largest genus of plants listed under that act. So the conservation of Prasophyllum, of the leek orchid species, is really of national significance in Australia. And when we're talking about, protecting and conserving Australia's biodiversity, we should be talking about protecting and conserving our leek orchids, amongst others, of course. But leek orchids have an additional problem to most threatened plant genera, and that's that we really didn't know how to grow them in cultivation. There have been a few successful germination trials over the last few decades, but they were few and far between, and the problem was that their seed just didn't germinate for some reason. So a quick refresher on orchid growing methods for conservation purposes. So leek orchids, like all orchids, have tiny microscopic seeds that can't germinate by themselves. In the wild, leek orchids rely on symbiotic mycorrhizal fungi to germinate their seeds. So we mimic this by going into the field, finding an adult wild plant, carefully extracting a piece of root from that plant, not too much because we don't want to hurt the plant, but just a little bit. Bring it back to the lab, cut the root open, and living inside all these coils of mycorrhizal fungi, we pull them out, put them on a petri dish, grow them up, and then add on our orchid seed. And usually when we add on our leek orchid seed, we get something that looks like this, which is not very much. And that's a problem if you're Prasophyllum tophanex from the Midlands of Tasmania, where there's only a single individual left on the planet. And I don't think there's any seed in seed banks for that species. I know there's no seed of Prasophyllum gilgai in any seed bank, and that's only known from 3 plants from a tiny reserve near Benalla in northeast Victoria. Hasn't been seen for 5 years. Prasophyllum correctum is only known from a couple of plants in eastern Victoria, and Prasophyllum fosteri from 8 plants. It's comparatively abundant from a single roadside in western Victoria. Now, these species are critically endangered. They will go extinct in the next 4, 5, 6-ish, maybe a few more years, but they're on the way out, despite our best efforts at managing them for most of them. The only hope these species have of not going extinct is if we get them into cultivation and reintroduce them, like Nushka was talking about this morning. They've done for other orchids very successfully. But we can't do that until we work out how to grow them. So we had some theories, of course, about what was going on with these leek orchids. We ruled out seed viability pretty early on. Seed viability is a bit low in some species, could probably be improved by cross-pollination, but it's really not low enough to explain our inability to grow leek orchids. And it's kind of a boring chapter in my thesis, so I'm not going to talk about it today. But the bulk of my study was focused on the relationship between leek orchids and their symbiotic mycorrhizal fungi. So first off, we started by little bit of a review type paper incorporating some unpublished data that's currently in press for the Annals of Botany, where we looked at what fungi associate, or what fungi do we find in the roots of leek orchids. It turns out that we find Ceratobasidium mycorrhizal fungi. So Ceratobasidium are one of the big three mycorrhizal fungi genera in orchids around the whole world. And leek orchids, interestingly, they associate with species of Ceratobasidium that are closely related to but different from known pathogenic fungi species. So some Ceratobasidium fungi are really quite nasty, agricultural crop diseases. Leek orchids don't associate with them, but they associate with their kind of slightly nicer brothers and sisters, which is interesting from an evolutionary point of view, how leek orchids have worked out how to harness the potential of these quite, aggressive group of fungi for their own good. It's really quite interesting. But of more interest to working out how to grow leek orchids was that this is Prasophyllum validum from Stawell in Western Victoria, and in a population covering about the size of a suburban house block, we found it associated with 4 totally different species of Ceratobasidium fungi. And that makes you wonder, well, in orchids that associate with multiple species of fungi, it's often the case that only 1 or 2 of those fungi species are involved in the seed germination process. And so maybe this could explain why we've been having such trouble growing leek orchids, because we might be sampling the wrong fungi and using the wrong fungi to grow them with. So to see if this was happening, I really did quite a deep dive into a few study species to see what was happening in the wild. First of all, what's happening in the roots of adult plants in the wild. So first cab off the rank was Prasophyllum lindleyanum, this beautiful green leek orchid, a really lovely plant. And in my study site, the adult plants associated with 7 different species of Ceratobasidium mycorrhizal fungi. 2 really common ones, present all year round. I sampled this population at 4 different times in the growing season. And 5 species that were only rarely found, only once or twice in my sampling. This is interesting. I wonder what species of fungi are associated with the germinating seedlings. So I made these glorified tea bags and filled them with seed, buried them around the base of the adult plants in autumn and watered them, because it never rains when you're doing a seed baiting study, and dug them up in spring and found all these beautiful germinating protocorms and seedlings. And you can see on this microscopic photo on the right, these cotton wool ball type white and sort of rusty orange. These are the coils of Ceratobasidium mycorrhizal fungus in this germinating seed, and they're feeding that seedling to get it to grow. It's a beautiful thing. So I mercilessly cut them up, those seedlings, and sequenced the fungi that was inside them, and found that from a heap of samples, every single sample was of this single mycorrhizal fungus species. So only one of the 7 species we found in the roots of the adult plants was ever found in the germinating seedlings in the wild. This could potentially, be evidence that the reason we've been struggling to grow leek orchids was because we're just using the wrong species. We weren't sampling the right one. So to see if this kind of theory was going to stack up, I looked at another species, Prasophyllum validum. This is that one from the house box sized population a few slides ago. Further sampling, showed that it associated, adults associated with 7 species of fungi again, and again only one of those 7 was found in germinating seedlings, although it was the most abundant species of fungus, so my theory was looking a little shaky here. A few new species were found in germinating seedlings, which was interesting. But our theory really fell apart when we looked at Prasophyllum frenchii, because that only ever associates with 1 species of mycorrhizal fungus in the roots of adult plants. And that, lo and behold, is the same species of mycorrhizal fungus that you find in the germinating seedlings. So there's really no clear evidence that sampling the wrong fungi is responsible for poor germination in leek orchids. It could be the case for some species like Prasophyllum lindleyanum, but it's definitely not the problem for Prasophyllum frenchii. So seed viability is not the problem. Fungi, probably not. The last theory we had was that it could be an issue with the germination media that we're using in the Petri dishes. So this kind of agar-based mixture. And the sort of like industry standard medium that we used for everything was this kind of oatmeal agar-based medium, like a glorified porridge, really. And everything loved to germinate on that. Calidanias, diurus, Thales, Thalimitra, Pterostylis, everything. But not leek orchids. And so I did a big symbiotic germination media trial with Prasophyllum frenchii and confirmed that, yep, OMA is pretty crap for growing Prasophyllum frenchii. But when I added in higher nutrient media or nutrient media with bran, wheat bran as the carbon source instead of the more soluble oatmeal, we did get consistent and reliable germination, and they maxed out about 5%. But that was a 5% germination rate with Prasophyllum frenchii. 5% is a pretty good germination rate, actually. And the seedlings grew really fast, not just in the Petri dishes, but also in the nursery, and started flowering 18 months after we'd sown their seed, which was really cool and really exciting because it was kind of we'd cracked the case, really. The nutrient composition of the symbiotic germination media was critical. And excitingly, we started to grow some really threatened Prasophyllum species. So we had a really good germination event with Prasophyllum fosteri, and we've now got a heap of Prasophyllum correctum seedlings in the nursery. So these species, these are two of the four that I mentioned up the top of this presentation, and they are less threatened, at least in the short term now, because we've got them into the nursery. We can seed orchard them and collect heaps of seed off these captive bred plants, and then we can use them for reintroductions. So it's really cool. We can now undertake breeding and reintroduction programs with a high degree of confidence for leek orchids because of this new method. So that's great. But I hear you say, well, that's wonderful, but come on, leek orchids look like grass. Who really wants to grow them anyway? What are the broader implications for orchid conservation? Well, I'm glad you asked. This is our slide from Prasophyllum lindleyanum a while ago, where we showed that adult plants associate with 7 species of mycorrhizal fungi, but only one is ever found in the germinating seedlings in the wild. And we actually did some ex situ germination trials using our newly found knowledge, actually using a different medium than what worked for Prasophyllum frenchii. Prasophyllum lindleyanum doesn't like that high nutrient media, but it loves the wheat bran, so that was good. But we found that every single species of fungus that we tested germinated in the lab, which is a bit concerning because usually for most of the species that we're working on in orchid conservation, we don't know this detailed information. We haven't done these seed baiting studies because they're really hard and expensive and time consuming. Usually all we know is what fungi germinate the seed. And so it's really important to ensure that we include a broad diversity of fungi samples in our reintroduction programs for threatened orchids, because if we don't, we could be missing some fungi that might be really crucial in the life cycle of the orchids that we're working on. And even for species that have very specific associations, like Prasophyllum frenchii, which only ever associates with a single species of Ceratobasidium fungus. This is the graph from our germination trial, where we used 3 different isolates, almost identical ITS DNA sequences, but they each preferred a different media. And the behaviour of them in the Petri dishes was quite different. So there's obviously some real diversity in the different isolates or different strains of fungi within a single OTU. And this backs up work that Celeste was talking about yesterday. There's this enormous diversity within an OTU, and different strains have different biology. It makes you wonder whether some are better for the orchids during drought years or during wet years. We really have no idea what these different fungi do, except that they all occur in the roots of the orchids, and there probably is some importance to them. So using a diversity of mycorrhizal fungi is likely important for threatened orchid reintroductions. This was a really massive team effort. I had help from so many people on this project. It was really, really incredible. So particular thanks to all the volunteers who helped me and provided me with information or collected seed and everything, my supervisors obviously, and the Threatened Species Recovery Hub, Herman Slade Foundation and Victorian Government. And I'd like to acknowledge the traditional owners whose land I did my fieldwork on. Thank you.